In the last session, we started to discuss about ear, and okay, so as a part of it, we have seen that external, middle, and inner ears, and we were discussing inner ear. And the inner ear has a cochlea, vestibule, and semicircular canals. And when we take the cochlea transverse section, so here I'm okay, so you can see the cochlea, the coiled portion of the inner ear is the cochlea. And when we take a transverse section and we see here, okay, so there are three channels, right, and are filled with the fluids. So here this is scala vestibuli, so we have seen in the last session, and it is filled with the perilimb. And again, this is a scala tympani, this is also filled with the perilimb, whereas the cochlear duct, so that is also called the scala media, so it's called scala media. So this scala media is filled with the endolimb. So it is filled with endolimb, and whereas this scala vestibuli is filled with the perilimb. So this is also filled with the perilimb. Okay, and here I told you that okay, so this will be the base portion. So when we see the coiled portion of the cochlea, so you will find like this. So this will be the base, okay, where you will find oval and round window, and this is the apex. Okay, so at apex there is a helicotrema. Now this helicotrema is a part of the apex region, and it's a location where the scala vestibuli and scala tympani meet. So they meet in this location, so that is in the helicotrema region. Okay, and one more point, the helicotrema will have hair cells, and these hair cells detect low frequency sounds, will detect low frequency sounds. Okay, and here at the base you have on the top will be the oval window, and okay, below it there will be a round window. So this is round window. So it is to this oval window connected is the stapes. So stapes connects to this oval window. Okay, right, and this oval window passes into the scala vestibuli, and then the scala tympani ends with the round window. Let's see straightening this cochlea. So how we'll see when we put this cochlea in a straight. Okay, so this is the tip of the cochlea, and then so inside we have the scala vestibuli, okay, and then tympani, and then scala media. So here I'm just mentioning, actually here when coming to this okay, the scala media, which is filled with endolymph. Below this we have is a basilar membrane. So situated on the basilar membrane the actual auditory structure, the one which receives the sound waves, that is called organ of Corti, is present on a basilar membrane. So organ of Corti is present on basilar membrane, right? And it's the organ of Corti that contains auditory receptors. So contains auditory receptors. So it contains auditory receptors. Okay. So the organ of Corti is present on the basilar membrane. Okay. So you can see here in this picture. So this is the organ of Corti. So this whole is the organ of Corti. So later I'll discuss about this in detail, and you can see from those hair cells the nerves arising, and all those okay together become the cochlear nerve. So it becomes the cochlear nerve. Okay. So again, coming back to this cochlea, so where I have stretched it, okay, means uncoiled this portion, and you can see this is the apex of cochlea. So this is apex of cochlea, right? And here is the location, what I call it the helicotrema. So already I mentioned it's a point where the scala vestibuli and scala tympani meet. So here is the one which I take the scala vestibuli plus the scala media. So in this location, and below this I'm going to take the scala tympani. Scala tympani. Okay. And here, so how we see here, okay, so on this top here you have is the oval window, right? And below this we have is round window. So this is the oval window, and below this is round window. Okay. And connected to this oval window we have is the stapes. So you can see here the stapes is connected at oval window. Okay, right? And this round window is covered by a thin membrane. So it is covered by a thin membrane, so nothing is connected to this, and it is movable, so the waves will come out through this right round window. Okay. So now the information, so right, so the vibration, so whenever the tympanic membrane receives the sound, okay, it vibrates, and the vibrations are passed on to the malleus, then on to the incus, and then to the stapes. So this stapes will be moving, okay, back and forth. So the stapes will move back and forth, and that generates, okay, waves in the perilimb, in the scala vestibuli, and in scala media, in scala media. Okay. So now these waves will pass, okay, in this way, and then they pass, okay, through the media, and then passing through this, okay, organ of Corti, and they will enter the tympanic membrane, and they will come out. Okay. Now the low frequencies, I told you that the scala vestibuli, where the scala vestibuli and tympani meet, the helicotrema region will have hair cells that detect low frequency sounds. So the low frequency sounds may go up to, okay, this point. So you can see So different parts of the cochlea will receive different frequencies. Okay, so some waves may go, okay, so up to here, and then they pass down into scala tympani, and then return back into return out through a round window. Okay, so this is how it will be taking place. So here where I show, so those vibrations will pass from scala, okay, vestibuli, then into media, okay, and then through this organ of Corti, so it is how it enters into scala tympani. Okay, right. So now let's see the structure of the organ of Corti. I hope now it is clear in the cochlear region how it is present. So here I have shown you the apex, where there is a helicotrema present and has hair cells that detect the low frequency sounds. So they detect okay, low frequency sounds. Right. So here I'm showing you the diagram of organ of Corti. So this organ of Corti has another name, so we can also call it a spiral organ, and this will, or this contains hair cells, and these hair cells are auditory receptors. Auditory receptors, right? And you can see here in this picture, so these are hair cells, okay, right, and there are inner hair cells, so these are inner hair cells, and we have here outer hair cells, and these hair cells will be having on the apical part. So when you see a hair cell, so this is how it looks, okay, and okay, from the bottom, so we see there is a connection with the nerve, so you can see the nerve innervates this. Okay, so this is the hair cell, and okay, on the apical part of this hair cell we will find a large number of processes. So those large number of processes are called stereocilia. So here you can see, so these are stereocilia. So these stereocilia are not same as cilia, so these are different from cilia. So here these are right called stereocilia, and these stereocilia will be in contact okay, with a elastic membrane. So this a thin elastic membrane called tectorial membrane. So you can see here, so this is a thin elastic membrane. So this elastic membrane is called tectorial membrane, and you can see here, okay, where he mentioned as a hair bundle is the stereocilia. So this is stereocilia, right? So here this is nerve fiber. It's nerve fiber. Okay, so this is the arrangement, okay, on the basilar membrane. So here down below the head cells we can see the basilar membrane. So this is the basilar membranes, and the hair cells are supported by a number of other cells, and here you can see all these nerves arising from inner hair cells as well as outer hair cells. So here the movement in the liquids will make, okay, the movements in these you know hair cells, so pressing against the tectorial membrane, which actually results in right generation of action potentials in the hair cells. So that is, I can say, bending of hairs. So bending of hairs in one direction, in one direction results in one direction results in depolarization, results in depolarization of hair cells. So that is, it results in the generation of action potentials which will pass through these nerves. Okay, so these nerves all, the nerves from the cochlea and the nerves from the vestibule, that I'll discuss okay, shortly. So here the nerves from cochlea and even the nerves from vestibule, so together they form vestibulocochlear vestibulocochlear nerve, forms vestibulocochlear nerve. So this is eighth cranial nerve. So this is a eighth cranial nerve. Right, so coming back to this diagram again, so here this portion which is present between the scala vestibuli, okay, and tympani, in the scala media we have is, okay, so the basilar membrane. So this is the basilar membrane, basilar membrane, on which present is the organ of Corti. Okay, so here I have already mentioned that how the sound waves pass. So here the base of the cochlea will receive high sound, I mean, high frequency sounds. So the base receives high frequency sounds. So base high frequency sounds, whereas the apex will receive low frequency sounds, and of course the middle portion will receive medium frequency sounds. So here say suppose this is the medium frequency sounds, and okay, and these are high frequency sounds almost to the region of helicotrema, and at the base they receive low frequency sounds. The base is meant to receive low frequency sounds, the apex high frequency sounds, and in the middle region it will be medium frequency sounds received. Now let's see how the action potential generates in these hair cells. So here, as I told, that when the basilar membrane moves, the hair cells will get pressed against the tectorial membrane, and the movement of stereocilia will result in depolarization of the hair cell. So here is the hair cell, and I told that, okay, on one side they have these stereocilia. So this is how the stereocilia, okay, are present, and below this is the nerve. So there is a synapse that exists between the hair cell and so this is a hair cell, and this is the nerve fiber, is a nerve fiber. And here we will see in the hair cells there will be, okay, calcium channels. So you can see here these are calcium channels, okay, and inside we find vesicles that contain neurotransmitters. So these are vesicles containing neurotransmitter. containing neurotransmitter. So these are stereocilia. Okay, right. And here the tip of this stereocilia will have ion channels for potassium ions. So here, when this, right, so those potassium ion channels are present at the tip of the stereocilia here. Okay, so here when the stereocilia comes, okay, pressed against the tectorial membrane, so here we have is the tectorial membrane. Okay, so when the stereocilia pressed against this against the tectorial membrane, so I'll just write TM, which means tectorial membrane. Now it results in the opening of these ion channels. As a result, the potassium ions will move into the head cell. So the potassium ions, they move into the head cell. And here the entry of potassium ions into the head cell. So entry of potassium ions into head cells, it results in depolarization. It results in depolarization of head cell. And this actually results in opening of the calcium channel. So here I told we have is calcium channels, and these, through these calcium channels, okay, so through these calcium channels, the calcium enters into the head cell and binds to these vesicles and makes these vesicles to come in contact with the, okay, the membrane, so where they fuse and results in the release of okay, so you can see here there is a release of neurotransmitter, so release of neurotransmitters into the synapse, and thereby, okay, depolarizing the nerve fiber. So this is how right, the nerve impulses will be generated, or the action potentials generated in the nerve fibers that are supplied to the head cells. Okay, so whenever the basilar membrane moves, the head cells will be pressed against the tectorial membrane, and that results in opening of these potassium ion channels through which the potassium ion enters into the hair cell, depolarizing the hair cell. That actually helps in opening the calcium ions and entry of calcium ions into the hair cell, and then binding that calcium ions with the vesicles and making the vesicles to bind with the membrane and okay, break open to release the neurotransmitter. So you can see here the neurotransmitter released into the synapse and thereby depolarizing the nerve fiber, and that information will be passed through this, okay, cochlear nerve right to the brain. Of course, the cochlear nerve will also receive the nerves from the vestibule. Together they become vestibulocochlear nerves. Okay, next we'll see about vestibular apparatus. Vestibular apparatus. So coming to this vestibular apparatus, so this will be consisting of semi-circular canals plus the otolith organ. So these two together will constitute the vestibular apparatus, and this vestibular apparatus is concerned with the maintenance of balance, and posture of the body. Maintenance of balance and posture of the body. Okay, Let's see here at the base of cochlea located is this vestibular apparatus. So we'll see. So this is cochlea here. Okay, and from here we have is the semicircular canals. So right, so this is how it looks. So this is the cochlea part. Okay, here you will find the oval window, and here is the round window. So this is oval window, and below this is a round window. And the locations of okay, so this region will be having, okay, utricle, and this is the region where we will find a saccule. So I'll come to that point. Okay, saccule, and this is utricle. So utricle and saccule. So that's the vestibule. So this is the one where, okay, in the membranous labyrinth, okay, the vestibule will consist of two sacs. So one that is saccule, and another one is called utricle. So this saccule will help in perceiving. So it perceives a vertical movement. So like when you go in in a lift, so that is perceived by the saccule. And coming to utricle, this one perceives a horizontal movement. So when we go in a bus or a car, so that movement is received by this utricle, and when we have a vertical movement, that is received by saccule. Okay, so this saccule and utricle together is called otolith organ. It's called otolith organ. Together it is called otolith organ. Okay, so I said, so this otolith organ plus the semicircular canals. So here these are semicircular canals. There are three semicircular canals. So three semicircular canals are present, so which will be perpendicular to one another, right? And this, as I said, the utricle and saccule together will constitute the otolith organ. Then the saccule and utricle, they contain a projecting ridge. So there is a projecting ridge, and that is called macula. And the macula have receptors for gravity. So macula have receptors for gravity. And coming to these semicircular canals, we have, as I said, three are there. Okay, so in this one is called superior right semicircular canal. One is superior semicircular canal, and another one is horizontal, okay, semicircular canal, and one more we have is a posterior semicircular canal. Okay, right. So this one is the superior semicircular canal. is semicircular canal, and this is the posterior semicircular canal. And this one is horizontal horizontal semicircular canal, is horizontal semicircular canal. So these are three canals, and this vestibule are all of these are filled with the perilimb. So they are all filled with the perilimb, and the base of these semicircular canals is swollen. So there is a swollen regions. Okay, so these are called ampulla. So here the base of semicircular canals are swollen, and those swollen parts are called ampulla and this ampulla region contains a projecting ridge. So it contains a projecting ridge, and it is called crista. It's called crista, and this crista has hair cells. Okay, and these hair cells are the one which receive the information. So they are receptors. So let's see how this crista is present in the ampulla region. So here, okay, so this is the projecting ridge, and here we have our hair cells. Okay, so just I'm showing a few here, and okay, so here these hair the stereocilia of these hair cells. So the stereocilia of these hair cells are embedded in a gelatinous cap. So here it is a gelatinous cap, and this gelatinous cap is called cupula and here already I mentioned. So these are hair cells and arising from the hair cells we have is nerve cells. So you can see here is the nerve cells. So this forms the nerve cells. Nerve fiber. Okay, so here this all I told you that inside the vestibule. So here this part is the vestibule part. So this is vestibule, and when inside the vestibule and the semicircular canals all are filled with fluid called perilimb. So here is perilimb present, and the movement of that perilimb will move the gelatinous cap, and when the gelatinous cap moves, then it bends the head cells, and bending of the head cells, we know it results in depolarization, and that results in generation of action potentials that pass through the nerve fiber. Okay, so this will be the vestibular nerve from these regions. So especially, okay, so here we have is the ampulla region, and here ampulla, and here is ampullary region, and this ampullary region have that crista which I have shown here, right? And this gelatinous cap will contain, so it contains right, small calcium carbonate crystals, so called So these are calcium carbonate crystals, right? And these are called otoliths, So these otoliths, we can also call them ear stones, and movement of them will also cause the depolarization in the head cells. So here, as I said, the base of the semicircular canals, it is swollen, and they contain that swollen part is called ampulla, and that ampulla contains crista. So of course its full name is crista ampullaris, and that has hair cells, and they are arranged in this manner. And here the nerve fiber, so this is the vestibular nerve. So this is the vestibular nerve. Okay, so this vestibular nerve, which will be right receiving the impulses from the semicircular canals and the utricle and saccule. So from here you will find those. Okay, so this is the nerves. So this will constitute the vestibular nerve, and from the cochlea we have is the cochlear nerve. From cochlear nerve. So now this vestibular nerve and cochlear nerve all, okay, together, so that results in formation of a vestibulo-cochlear nerve. So this is vestibulo-cochlear nerve. Okay, so plus cochlear nerve, so that constitute vestibulo-cochlear nerve. Right, and one more point here. So here, as I mentioned, that the saccule and utricle have a projecting ridge called macula. It is the macula that will contain these otolith organs. Okay, so here I'll just mention that. So crista ampullaris. Okay, so its gelatinous cap. So its gelatinous cap do not have otoliths, whereas the macula, so which is the projecting ridge in saccule and utricle, so its, okay, gelatinous cap contain otoliths, right? So here we have other names for these otoliths. We can also call statoconium, or, okay, otoconium, or also called statolith, are different names for these otoliths, which are calcium carbonate crystals embedded in the gelatinous cap, especially in the macula, which is a projecting ridge present in saccule and utricle, and whereas the projecting ridge of these semicircular canals, that is crista ampullaris, will not have these otolith organs. Okay, so that's all about the ear. So in the next session we will see about nervous system disorders, and after which we will start discussing about objective on this neural control and coordination.